a lot of the different functions of all these little pieces that are inside the cell. And the first thing I want to do is I want to talk about the role of the nucleus. So we're still in chapter 7, which is all about the cell. And what I want to start today off with is a discussion of the nucleus. So what do we know about the nucleus? What are the facts that we know?
Well, is it the nucleus, or are we talking about the nucleoid region?
What we'll assume from this point onward is we'll just assume that we're all about eukaryotic cells.
Okay.
So tell me about the nucleus. Okay, contains DNA. Which we know is the genetic material. What do we also know? Okay. It has a double membrane. That's good. The term that we might use instead of double membrane from this point forward is we might say that it's a lipid bilayer. What type of lipid are we talking about here? Perfect. What else? What else is inside of it aside from just DNA? It'd have to be RNA because it's the site of RNA synthesis, right?
What synthesizes RNA?
What synthesizes RNA?
Yes.
A couple different enzymes. The name of the biggest or most important enzyme would be RNA polymerase,
Okay.
which makes the polymers of RNA.
Okay, and does that enzyme live in the nucleus?
Mm-hmm. Operates in the nucleus. We talked about this the other day. You had written down on the last part of your chapter reading guide for chapter 5 mostly, you had written down that RNA synthesis occurred primarily in the nucleus. You wrote it down as transcription. It was the very last question on the back. And through a nuclear pore, there are things that can enter and there are things that can leave your cell or your nucleus. The nuclear lamina, which is basically protein that works to maintain or helps to maintain the shape of the nucleus.
Hey, Mr. Fields, I don't see a C drive under my computer. Should I just make a folder or what?
You don't see C drive at all?
No, I see K, N, Q, R, and Z drive.
Put it on the desktop.
Okay.
Okay, what else do we know? We know that inside of that is the DNA. We know that DNA is located on chromosomes. So I want us to know that DNA and protein come together to form that chromosome.
I have a question about that.
Mm-hmm.
So when it's just kind of chilling in the nucleus, is it in the form of a chromosome or DNA?
Well, it kind of depends on what area of the chromosome we're talking about and what time frame the cell's in. You could imagine that if you were reading DNA the best way to read it would be when the DNA strand is lengthwise written out, correct?
Mhm.
However, this is good for reading, but it's not good for maneuvering it and moving it around. Right there is just DNA. Now, there are certain proteins that will then associate onto the DNA. So right here I'm doing these dots, and these dots are proteins that are going to come and act on the DNA. And what they'll work to do is they'll start to fold the DNA.
It's like that video, right?
Yep. An elaborate shape, and we call that a chromosome.
Okay, so for instance, like it was during,
Like
cell.
when the cell's going to split,
Yeah, then it
like
would be,
that would be analogous to like you moving from house A to house B, or taking like all of the contents of your house and then moving to two separate houses.
So then you want that in a chromosome.
You would want to package all your stuff in a box or some kind of crate rather than moving it individually piece by piece, because
Okay,
that's
then
very
during
tedious.
Transcription, it's just
So in transcription, when you're needing to read the DNA, you would have it lengthwise like this.
okay, and when is in the chromosome, is it a double helix inside of the chromosome?
Yeah, so like the DNA that is making up this like part right there, you can imagine this blue line. If you were to zoom way in, I can do that right here. So if you were to zoom way in, you would see that it's your double helix, if you will.
Okay, thank you.
Mm-hmm. Let's talk about something else that's in the nucleus. It's an area of the nucleus called the nucleolus. It's an area, or maybe we could say the term region, within the nucleus. That makes our RNA. Now we know what RNA is, right? It's like DNA, but only not, it's like long term. We can think about it. The R in front, though, is maybe a little new. You've seen, though, the M before. What did the mRNA stand for? What did the M stand for? Messenger. The R here is going to stand for ribosomal. So this is going to be RNA that is important in forming the ribosome. Cool. The next thing I want to talk about a little bit are those ribosomes. What are ribosomes made of? We know one thing that a ribosome is made of. What was it? We just established this fact. Made of our RNA. From our reading, we might have seen that it's made of something else as well. Very good. And there are two pieces, two subunits that make up each ribosome. There's the small subunit and the large subunit. And together they come and they coalesce to form your overall ribosome structure. So I'm going to write down a ribosome, which is again made of these two pieces. And along the way, the mRNA that is being read by the ribosome runs right through that connection point of the two. So you can imagine what happens here. Imagine my hand is a ribosome right now, and this is the mRNA. The mRNA is put right in here. The other unit comes on top, and we start reading it. You ready? Illustration. Start reading the mRNA. You understand? Basically. Or another example. Y'all seen this before? I had a student make this for me a couple years back. Try to get this on the camera. That's good enough. So there's my ribosome, right? I can pull this out for now because we don't need this just yet. Or at least we don't need to see this on the screen. Okay, so here's the large subunit. Do you see this little division right on bottom here? You can kind of see this white line. There's the small subunit. And the two come together is this, which is a strand of, you see what that says? mRNA comes through. And you start reading the mRNA one piece at a time. Now realize here that there's no A's, T's, G's, and C's, but you can imagine that there are, right? Or it's actually A, U, G, and C because this is RNA. So you start feeding it through after the two come together. The one problem is that sometimes this doesn't line up. Oh God. And this obviously doesn't work out that well, but you can understand like what's trying to go on here. It is pretty creative. It just malfunctions a little bit sometimes. Oh well. Y'all get my drift, right? It's got to feed this through. It's not feeding right now, whatever. And look what I start to make. This is a protein chain, right? Do you see that? That says CYS, that's cysteine. There's lysine, proline, serine right there. So there's all the amino acids that are forming the long polypeptide chain. What do we think? Pretty nifty, huh? Doesn't really work that well, but it's pretty nifty nonetheless. It is kind of cute. Okay, more on that later. Ribosomes could be found in two spots. There's two places. And just to make sure we understand, what is a ribosome doing? What's its overall function? Okay, so overall function is to make protein. And it's found in two spots. What's one spot?
Outside the nucleus.
Well, definitely always outside the nucleus. One spot is where it's attached to something called the endoplasmic reticulum,
Oh yeah, the rough ER.
which makes it the rough ER.
The gold, what you got for
It comes later. Comes later. Sometimes they're also just found free in the cytosol. What was the word cytosol from yesterday? That was the liquid, right? Cytoplasm was the area. So they're found free in the cytosol. Now, if you think about it, their overall function is to always make proteins. So here's the way that this works out. You can see that there's two places you can make these proteins, which are where the ribosomes are at. Either endoplasmic reticulum based or free in the cytosol. If a protein is produced on a ribosome that is free in the cytosol, that means it's for that cell. It's the cell's protein. If it's made on the endoplasmic reticulum, though, the rough ER as we call it, that means that the protein's probably going to be exported. The protein would be exported. What does export mean? Sent away, right? If it's for some other reason. Okay? Let's talk about the endoplasmic reticulum real quick.
I think that is.
No, it's not.
No,
It's
it's-
like 9:45.
There's no way that could be Osbel.
Yeah, we do have to leave.
7 minutes, 8 minutes left. So let's talk about the endoplasmic reticulum. There's two types. There's the smooth type, so the smooth ER. There's also the rough ER. What's the smooth ER doing? Well, first off, why smooth versus rough?
The rough has the ribosomes on it, and this moves to the
So no ribosomes here. Yes, ribosomes here. Correct? So which one of these do you suspect is all about protein something or other? The rough, correct? So I'm going to say that this is all about like protein synthesis or some level. Protein.
Teachers, please excuse that bell and we still remain in class. Thank
I'm
you.
gonna write protein base there. We'll see how far that goes. Now let's take smooth ER and let's look at and see what it's doing. Obviously, it's not really all about functioning to do anything with proteins. And there's actually like 4 things that the smooth ER does. One of the things that it does is it detoxifies poisons. Another thing that it does is it metabolizes carbohydrates. It also works to store calcium. And also is a site for lipid synthesis. Did we see anything with like proteins in that listing of smooth ER actions? No, because it is smooth with no ribosomes whatsoever. Good? Now moving on to the rough though, obviously that's going to be more protein based. And here's the deal. If you remember from before, I had said that if you had ribosomes that were attached to endoplasmic reticulum to make it the rough ER, then those were proteins that were typically exported from the cell, right? So here's the way this works out. I'm going to try to draw this real quick. We'll see how far it goes.
And is it shaped like that to maximize the surface area?
Yes, to some degree. Surface area is not as pivotal though for ER as it is for other organelles, I think. Do you know what I've just drawn right there? A ribosome that's attached to the ER. So I'm going to write ER here, and I'm going to write ribosome right there. Now the ribosome is reading what? What does the ribosome read? Specifically mRNA. So that it can produce a what? Protein. So a polypeptide sequence, right? So here's my polypeptide sequence. What is each of these black dots representing? So there's 5 amino acids in my polypeptide chain here. Now, the next thing that happens is that polypeptide chain that you're seeing right here is fed into, and this is almost automatically, into the ER. So now, your polypeptide chain that's actually being produced is being fed almost immediately to the inside of the ER, the ER lumen as we call it. So in the end, you're left with a polypeptide chain. I'm going to draw just a little line right there and write the word polypeptide. So we can imagine like a little bit of a time-lapse thing. We have a polypeptide right on the inside. Cool? Now it's in here in this rough ER that this protein-based like production center is actually taking and it's modifying the proteins. So in your rough ER, its overall function is to modify newly produced proteins. Some of the modifications can include folding, protecting it, facilitating its transport. There's a lot of things that are going on to help facilitate this protein. So let's think of information travel so far. We started out in the rough ER, and now where do we go next?